Is that better?
Yeah.
Okay. 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 So, um, test case, thinking about humans adapting on these rapid timescales, infectious disease being one of the fastest, strongest pressures. We look to the islands of Cabo Verde, and this is a study or a series of studies led by a former PhD student in my lab, Iman Hamid. So I made this claim before that host biology and genetics are key factors in disease distributions and severity. And one of the classic examples of this is a single point mutation that changes the expression of this gene. And we call it the Duffy negative mutation or allele. I'll refer to it a few times like that, which is highly protective against the malaria-causing parasite Plasmodium vivax because it turns off expression on the surface of red blood cells of the main antigen that is used for the parasite to bind to invade. So if the parasite can't get in to the red blood cells in humans, a lot of the downstream disease consequences are severely dampened. So because of this clear benefit, this has swept through African populations and is rare outside of African ancestry. So this is a classic example of rapid adaptation in human populations. We're looking at just plots of the allele frequencies here. So really high frequency of this adaptive allele in blue here. But if you fill out the rest of the geography, you can see that there's substantial amounts of blue, this adaptive allele frequency in other parts of the world. And this is primarily through a process that we'll refer to as admixture, where you have multiple source populations contributing to admixed population. So in this case, there is substantial amounts of African ancestry in the Caribbean populations and other American populations. And remember, in most African populations, there's a really high frequency of this adaptive allele. So it came over during the transatlantic slave trade on that African ancestry. And more generally, admixture leaves specific genetic signatures in our genome. So we have a little cartoon here where we have two populations, and these genomes are just colored from the population that they were sampled from. And if you combine the gene pool, then over time, recombination will break up these tracks. You can scan along the individuals in the population today and say this track traces back to population 1 versus this track traces back to population 2. So our hypothesis on the islands of Cabo Verde, who have ancestry from Portuguese colonizers and enslaved West African people, is admixture facilitated adaptation to malaria via the African origin Duffy negative allele. So we use Cabo Verde as a case study for a few reasons. So this is the island. Oh, it's really light on here. You might be able to see it better on Zoom. But data collected by our collaborator, Sanjay Beleza, we have about 500 some individuals across genotyped at a little under 1 million SNPs. And we want to focus on the islands of Santiago. So there's a bunch of islands there. You don't really need to see it. But, why Cabo Verde? Why this setup? So we're interested in this specific phenotype of resistance to Vivax malaria. And when people reached Cabo Verde in the 1400s, they brought malaria with them, but only to one island primarily, Santiago. And one of the more fun things I think we've done is we looked in captain's logs from the 1700s, and there were descriptions of periodic fevers, which we associate with being malaria, that people encountered. But notably, they found differences in the severity of the disease and the fevers between African and Portuguese populations. And this matches what we know about the Duffy negative allele and supports the fact that this would be malaria. So we think that Cabo Verde is really an ideal case study to test our question of kind of adaptation to disease in action on kind of epidemiologically and ecologically relevant timescales for a few reasons. There's a strong time limit for the onset of adaptation because the islands were uninhabited before the 1400s. So normally when you're doing population genetic analyses, you're kind of jointly inferring the timing and strength of selection, and there's some like complex interaction between the two and like identifiability issues. But we know from historical records a lot about these populations. There's also, as close as you can get in empirical human populations, natural controls, where you have multiple island systems with a lot of shared demography and ancestry from both of these populations, but only one really has established malaria. So you have the selection versus demography that you can maybe tease apart. And importantly, malaria in particular, because we kind of think if you can see adaptation on this time scale anywhere, you can see it here. So how do we go about looking for selection in the population of Cabo Verde? Well, classic methods to detect selection from genomes are not really applicable to admixed populations. So we usually think of a whole bunch of haplotypes. So each of these is a chromosome sampled from an individual. So a whole bunch of individuals. You have an adaptive variant that's at really high frequency because it's beneficial. And you have a really long haplotype because it went up to frequency really quickly through adaptation. And so there wasn't a lot of time for recombination to make that chunk shorter or for new mutations to be introduced in the nearby region. So we have the signature of high frequency allele, long homozygous haplotype around it. But then you have admixture from a population that didn't have adaptation. And now both the allele frequency was diluted and there's neutral mutations introduced nearby. We can't do our normal selection scans that well. But there's reason to be hopeful as well. There are both biological and statistical reasons that admixed populations are kind of a great test case to think about rapid adaptation. So on the flip side, you can't use those common methods, but you can think about a few other methods. So we have the same scenario where we have adaptation in one population. Here it's blue, adaptive allele star, and no adaptation in the orange population. You have recombination creating a new population here, admixed population, just by chance, no selection yet. Because there's, say, 30% blue ancestry in the population, we expect around 30% of individuals to have the adaptive allele. Again, no selection yet. But that means that you have a high starting frequency. It's not like a new adaptive de novo mutation that starts at a frequency of 1/2n. That's really likely to be lost stochastically due to drift. It's, at 10, 30% frequency. That's not going to be lost randomly. If the allele is advantageous in the new admixed population, like it was in one of the source populations, then as it rises in frequency, that same adaptive process, it brings with it a new statistical signature because it's only on the background of blue ancestry that as the allele rises, blue ancestry in that local region rises as well. So we have a new suite of methods that we can use where people think about ancestry-based signatures of adaptation. And the most common way to do that is to break the genome up, usually by like single loci or chunks, and scan along to create an empirical distribution of the background amount of ancestry by locus. So a histogram of this locus has about 50% blue ancestry, and this locus has 75% blue ancestry. And then you compare where in that distribution is your adaptive allele. The hypothesis being that it will be an outlier with more ancestry from the adaptive population than the rest of the genome, which gives kind of like the neutral background of what demography produced. So what does that look like for Santiago? This is the empirical background. And the region containing the Duffy negative allele is the single highest proportion of African ancestry in the population. And notably, remember somewhat controls? That's only true in Santiago. It's not something unique about the region, something about the recombination map or anything, because it's well within the range for the other island populations of African ancestry. But a nice simulation study done by a former postdoc in my lab showed that these ancestry outlier methods, as they're often called, are both underpowered and prone to false positives. I won't harp on that too much. So that was really an impetus for us to think about other information that we get. So there was kind of the shift in population genetics where we went from using like allele frequency based methods to study selection to haplotype based methods to study selection. And since we're at UCLA, like John November, who used to be here, is really one of the pioneers of these haplotype-based methods. And so we thought about kind of the analogous version with, instead of just looking at ancestry proportions, looking at the spatial, like the length of the ancestry tracts. And so it's not just about a high frequency of the ancestry, but how long on either side does that orange ancestry go? Because it's a kind of information about how fast the allele rose to high frequency, because alleles just drifting can also rise to high frequency, but they'll do it much slower than an adaptive allele. So they'll have more recombination events to make that tract shorter and shorter. What does that look like in the data? We have a whole bunch of individuals stacked on the y-axis here, and then the entirety of chromosome 1 on the x-axis. So that's for perspective, like a huge chromosome. And we still have ancestry tracts after 20, 25 generations that span almost the entirety of chromosome 1, which is really unexpected. So these definitely support the idea of selection. You'll notice that there's a lot more higher proportion of West African ancestry in orange than blue. That was at ancestry outlier detection, but they're also much longer. And we quantify that by looking at like the decay from the dashed line as our adaptive allele. As you go out, how rapidly do you switch? And this is a very similar kind of concept if you're familiar with things like IHS statistic for non-admixed populations. But this is really hypothesis driven about a specific locus. We also wanted to do a more formal test, especially thinking about a broader class of questions where you don't necessarily have a hypothesis. And so we turned to developing a new method using CNNs on images of genomic windows to make inferences about not just the presence or absence of selection, but localizing it within the window. So it's a similar, we did a similar approach where we take two populations, this is all simulation-based data, and have one population with an adaptive allele, one population without, and generate these images that are basically painted ancestries, where you have a whole bunch of individuals stacked on the y-axis, and you are spatially going across, say, chromosome 1 on the x-axis. And you can kind of squint and see, like, where would selection be in this scenario? Well, these object detection methods put down a bounding box that says right here. So it's the signature of there was a lot of high frequency, like a lot of black there. So it's across most individuals have that high frequency allele, and then they're like longer tracts as well. So it's a very similar type of information. And the underlying architecture is very similar to these object detection methods that you may have seen before to look at images. So we compare kind of our different methods. We can see an ancestry outlier methods. Again, this is the entirety of chromosome 1 on the x-axis, and then the ancestry proportion. So we're looking for like outliers from here, and it calls a few different hits in that general region of Duffy. The true is this like window in red, and then, but it's kind of across a broad space with multiple hits. So ancestry outliers do okay here. We know like if they're going to perform anywhere, they perform in this really extreme example, but it's still, if you didn't know what you were looking for, hard to figure out how to interpret this when you have like 48 megabases to look over. Versus the object detection has the option to throw down, multiple bounding boxes, localize a lot, but it was a little hard to figure out how to plot it because the identified region overlapped so exactly with like the true region that they just looked the same. And so you have a lot. If you want to do functional follow-up, it's a much smaller region. Simulation-based approaches estimate it to be about a third the size. So you have way fewer genes if you want to do kind of functional follow-up to figure out what you're actually doing. I'll mention there's a lot more in the paper about validating this approach and where it does and doesn't work well, but I will kind of skip over that. Happy to take questions about it. But going back to our actual theoretical question, or our phenotypic question that supports the idea that there's selection at this locus, but it doesn't tell us anything about the kind of evolutionary parameters or the model. So for that, we turn to approximate Bayesian computation, which is a way to infer the parameters, where we have this a variety of summary statistics, for example, those lengths of ancestry tracts, the frequency of the allele that are informative in some way of the parameters that we want to infer. So things like selection strength or the timing of selection. And these show a correlation because the summary statistics are informing about the selection strength. They tell you something about that. So you do a whole bunch of simulations where you know the parameters, and then you compare where does our data fall in that, plus a little bit of error, to get a posterior distribution or best guess estimate for these parameters. So using our data, we use just a flat prior over a huge range of selection coefficients, and we get a nice posterior that looks pretty different from the prior, so you kind of trust that there was enough information there, and that's centered on a selection strength of about 0.08. It holds for a variety of starting frequencies and dominance. Kind of theme in my lab over the last few years is how do we not already know this about malaria, given we've been studying it for so long? Like we don't know the dominance coefficient of Duffy negative, even though it's been known it's a classic locus for over 50 years, somewhere between 0.5 and 1. But to kind of put this in context, it is one of the strongest examples of selection in a human population. And kind of epidemiologically, it would have decreased the pool of Vivax susceptible individuals by about half over the last 500 years. And this really tracks where we have evidence of Vivax on the founding of Cabo Verde, the peopling of Cabo Verde, but there isn't really Vivax on the islands anymore. There's occasional falciparum outbreaks. Cabo Verde is actually the most recent country to be called malaria-free. But Vivax hasn't been on the islands for quite a while, and it's not surprising given that very few people are naturally susceptible anymore. Okay. Okay. So going back to our initial questions, what ongoing pressures do pathogens exert on human genomes? Admixture facilitated adaptation to Plasmodium vivax malaria, which dramatically changed both allele and haplotype frequencies in Cabo Verde over the last 22 generations. But you can also think more broadly. So one of the kind of questions that we have is, okay, adaptation to infectious disease is crazy fast and crazy strong. This is on the scale that, as I said, ecologically and epidemiologically relevant, hundreds of years. How does that affect the genome? Because this is a regime that we're not often thinking about. So we did a variety of simulation tests to see kind of the bias towards demographic inference and kind of genome-wide patterns of ancestry. So we have the same setup. This is the setup for the simulation. We have one population with the adaptive allele, one without, and then evolution, including adaptation at that allele. And then we plot for different selection coefficients that we put in for the strength of the, I'll do it in here, for the strength of the adaptive allele. How does global ancestry change after 20 years or 20 generations or so? So we break it into two different sets of chromosomes. One is chromosome 1, which is where the adaptive allele is. So it's not surprising that we know the ancestry in chromosome 1 was pushed higher towards African ancestry because the adaptive allele brought these huge chunks of African ancestry with us. But what surprised us is even if we throw away chromosome 1, the rest of the genome also has this strong relationship between adaptation, introducing more African ancestry. And that's because our kind of classic way to think about adaptation, we're like, oh, well, if we choose regions that are to infer demography that are far enough away from selected genes, we'll be okay. They're like functionally neutral, right? Well, if adaptation is this fast and strong, you can't decouple from the pedigree that quickly. Like it's really a different selection regime where recombination doesn't have time to decouple the impact of selection at that single locus from the rest of the genome. Because if you have African ancestry at the Duffy negative locus, chances are you have it in other parts of your genome as well at a higher frequency. So those whole individuals and their whole genome are more likely to reproduce, even though they're physically like uncoupled within the genome. So then selection on a single locus can influence genome-wide ancestry, and inference is important to think about the impacts and biases for demographic inference as well. More generally, we think that methods to detect admixture-mediated adaptation can be used to study human and non-human populations, and we look to work with other people to use our methods in other scenarios. One of the nice things about these ancestry-based approaches is that there are increasing number of tools that can be used on kind of low coverage or poor quality data to infer local ancestry. So for things like non-model organisms or ancient DNA, you can often do a much better job at inferring local ancestry tracts than you can do at hard genotype calls. So you can use these types of methods much better than you can use kind of some other types of methods. And papers are down there for more information. But kind of a broader perspective is we often think admixed populations are an ideal system to study rapid evolution common in infectious disease. And if you think about human populations kind of across space and time, depending on the time slot or the geography that you're thinking of, basically all human populations are admixed in some way. Even populations that are canonically thought of as homogeneous, like Europeans, have wide scale gene flow events replacing up to 90% of the population with the rise of farming, or around 50% of the population are in the Pontic-Caspian steppe migrations about 4,000 or 5,000 years ago. So there's a lot of opportunity to study this. a few reasons to think this, both kind of biological and statistical. Ancestry patterns are informative about population history on shorter timescales because the recombination clock versus the mutation clock. Admixture may introduce already adaptive alleles, so you're not waiting around for that slow part of adaptation, which is the de novo mutation. And these alleles are introduced at a higher frequency, so they're less likely to be lost stochastically due to drift. Okay. So that's kind of the first section, humans. It's also published work, so a little bit more established. And now I want to shift in kind of tone and a little bit to think about some of the more recent work that we've been doing in the lab, which is the other side of the story. So we're starting to do a whole bunch of sequencing of non-human primates and their parasites. And we're like, well, how do we analyze all of that data? We've been reading a lot more parasite, like empirical genomic papers, and trying to interpret some of the figures and saying like, I'm not sure if this is really the way that we would approach it coming from more of a population theory background. And so this is kind of the early stages of that line of thinking that we're going at in a few different ways. So, and part of this is led by a former postdoc in my lab, Malika Gopalan, who is now a professor at Clemson. And indeed, kind of over the last few years, we've really made major progress on in phylogenomic methods to interpret genomic data from viruses, really accelerated by COVID. But recombining parasites with complex life cycles have been left behind a little bit. So what is the expected neutral genetic variation of a malaria parasite, and why should we care? More generally, we now have tens of thousands of samples of whole genomes of malaria, really spearheaded by MalariaGen. And how do we go from genomes to understanding of basic parasite biology or public health? How do we analyze all of this data? Kind of the classic shift that happened in human genetics, say 20 years ago, where we've gone from data limited to methods or analysis limited. And this is kind of pressing because genetic epidemiology is already being used in the field, for example, to track transmission or the frequency of drug-resistant loci. But lacking kind of the underlying theory and population genetic methods, I think really prevents formal hypothesis testing and biological insight. It's much more descriptive at its current state. And so we also think that a couple empirical puzzles can be solved with a new framework that can combine epidemiology and population genetics, but that's a little bit further down the line. So what's the expected neutral genetic variation of a malaria parasite? And this is really important because neutral variation is really the basis for all adaptability and influences vaccine efficacy that we know about in the field. So there's a few ways that people have approached modeling malaria. So they're kind of ecological or epidemiological style models. These are SIR style models. And I think I saw Sally online. This is actually a little schematic from the paper that we wrote together many years ago. So shout out to her. But these are commonly going to assume that evolution is slow enough to be approximated as constant on these epidemiological and ecological timescales. Not all models do that, but it's fairly common. And we saw even in humans that might not be possible, and that's especially not true for the parasite. Okay, so maybe population genetic methods work a little bit better. Well, the complex life cycle of the parasite really violates most common population genetic model assumptions. So you have this dramatic bottleneck from transmission mosquito to human and then human to mosquito when you're talking about on the order of 10 parasites. And then you have asexual reproduction that it's on the order, like gets from that 10 to maybe 10 to the 8, 10 to the 9 parasites within a single host. And in the vector, you have a diploid state where the rest is like haploid, and you have sexual recombination possible and something on the order of 10 to the 5. So you have this dramatic change in population size over the course of a life cycle. So what does that do to interpretation of common genetic summary statistics when we read a paper and they scan along the genome and calculate FST or Tajima's D? Like, how do we actually interpret that? So we developed a 4-stage model of the malaria life cycle based on those 4 stages that I just mentioned, under the kind of basic premise, or not basic premise, an idea stolen from coalescent theory, where without doing a full coalescent model, when you have a population size that changes in time, the overall effective population size is the harmonic mean of the population over time. And so we can write out the harmonic mean. So that's this equation over here to get an estimate of the effective population size, which is a measure of genetic diversity in the population. So we have 4 different stages that we consider. We have the transmission bottlenecks, so human to vector and vector to human. So there's 2 of those. The times are on the top, like how long each stage lasts, and then the sizes of that population. So this is going to be on the order of 10 or 50 or 5 for these 2 bottleneck sizes. So that's those part of the life cycle. And then we have the within host and within vector exponential growth from bottleneck size to what we call C parameter of how large those get, which is going to be on the order of like 10 to the 8 or 10 to the 5. And they're all weighted by the amount of time that you spend in each of these different life cycle stages. So you can do a lot when you have an equation actually written down. And I think one of the most helpful things is to think about like elasticity and sensitivity. And we can look at a contour of how effective population size, which is the different color bands here, changes with different key parameters. So here we vary the bottleneck size on the x-axis versus the peak size in humans, like parasitemia. And you'll notice that the parasitemia, so that max carrying capacity in humans goes from like 10 to the 8 to 10 to the 12. So that's a huge range of multiple orders of magnitude. And if you like look at the bands, you cut them across this way, like it doesn't really change effective population size that much. Whereas bottleneck size, you go from like 1 to 5 and you're getting an order of magnitude difference in the effective population size. And this is well known of how harmonic means operate, where if you think about a bottleneck, it's going to have an overwhelming impact if you're adding, say, 1 over 10 bottleneck to 1 over 10 to the 8. So that's good, consistent, math is mathing. And importantly, despite having 10 to the 8, 10 to the 12 potential parasites in a human host, and then say, and millions of infected hosts at a time, we're still getting per host infection and effective population sizes of around 100. And then you can add in some metapopulation structure with like a right island model where this is like the number of infections. There are more complicated ways to like add in different hosts, but the per host is around 100, despite having, 10 to the 10 parasites there. You have a tiny amount of variation. So why does this matter? Sketched out a few different ways to think about it. One is kind of our sanity check. There have been multiple ways that people have tried to estimate from empirical data effective population size, but they all measure different parts of the life cycle. And so we can see that, for example, just confirming our prior results, if we do some simulations, the number of transmitted parasites affects pairwise diversity substantially more. So the difference between, say, 5 and 20 parasites, 4-fold change, has a higher impact than changing, 2 orders of magnitude of the within host parasitemia, like carrying capacity. Good check. Why does that matter? You can overinterpret this a little bit and maybe tell some stories. Something like treatment, which reduces the overall number of parasites in a host, may not substantially alter genetic variation. And again, this matches PopGen theory of this harmonic mean. But it also means that when we're doing genetic epidemiology to trace infections, it might be okay if we're doing certain treatment, depending on the types of summary statistics that we're using, if it's not altering genetic variation too much. We can also do things like reinterpret common summary statistics. So I mentioned we were reading all these empirical papers and they scan along the genome and calculate these common summary statistics that have a really good core theory developed around, say, Drosophila or humans. And Tajima's D is one of the ones that seems to be very common in empirical studies of malaria. And it compares 2 different estimates of kind of summaries of genetic diversity, 2 different ways to calculate effective population size, pairwise differences, where you have to compare between 2 different parasites. So under the sampling scheme of malaria, you usually take like 1 parasite per host and compare them for pairwise differences. And then the other is the number of segregating sites. So just overall mutations that you're seeing. Almost all of the empirical examples have this highly negative Tajima's D, and it's often interpreted as a recent expansion associated with the host shift from great apes to humans, and then because a host shift kind of leads to a bottleneck and then a massive explosion. But we did some simulations and calculations under our model with a completely constant host population at equilibrium. And we see even in that case, we get, this is just kind of a position on the genome on the x-axis, genome-wide average that is quite negative. It ranged from like 0.5 to, or like minus 0.5 to minus 2. And we think that this is really driven by the sampling scheme and the life history of the parasite, where you have the successive bottleneck and expansion in every generation, you have that expansion. It's not that you need to have a population expansion of the host, because you have that expansion within each host itself. And the sampling scheme actually exacerbates that. So it's not just a change in population size of the parasite over time. The way that we are pretending that this is a single population that we're kind of randomly sampling that is well mixed is not actually true. This is a metapopulation of hosts. And so you're dividing up the coalescent structure, which affects these two ways to infer diversity that Tajima's D is measuring differently. So we have two different stages. One is you can have everything that happened before the host you're currently in, where you're allowed to have between host coalescence, and they can shift between. So that's kind of that top structure. And that's governed by like the number of hosts and epidemiology. But then because of the way that we're sampling, where we have one parasite per host, and then comparing across a bunch of different hosts, we create these really long kind of tails or branches within the coalescent tree where you cannot have coalescence between different hosts because all the mutations happen only within that single host. So you have these really long branches. You had this bottleneck, so there's like a whole bunch of coalescence, and then huge mutation size or huge population sizes in the host where most of the mutations are reintroduced because you have like, say, 10 to the 10 individuals. And so you have these really long single branches with a whole bunch of singletons that are basically enforced by the sampling scheme. And so the two different parts of Tajima's D, that pairwise difference minus segregating sites, are measuring two different, or like sensitive to two different parts of the coalescent tree. And pairwise difference is you need to have coalescence between the two individuals to be able to see them, whereas segregating sites is going to be sensitive to all these singletons. And so it's not surprising that Tajima's D is negative. Are we on time? Okay. And then the last kind of vignette of why this life cycle model is interesting is a little bit more inside baseball, but I think it is something that I've been thinking about a lot myself lately. So we are not the first people to think about how the life cycle matters to malaria, but other pathogens as well. There was a really important paper towards this goal that came out a little over 10 years ago. And one of their main takeaways was that around 25% of genes in the Plasmodium and Falciparum genome that they looked at have pairwise diversity of non-synonymous, so pi n, to synonymous, so change amino acid versus doesn't change amino acid, of greater than 1. And this is really surprising compared to, 2 to 5%, 2, 3% for organisms like humans and Drosophila. Because this ratio of pi n to pi s is often a measure of different types of selection. So it's usually going to be driven by purifying selection, driving this to be like on the order of around like 0.2 or 0.3 genome-wide, this average, because most mutations that are non-synonymous are breaking something that's working pretty well. So they're going to be pushed out. And then synonymous is like roughly drifting neutrally. And then you have on occasion something that's really great for the genome, beneficial allele, non-synonymous changed, made it great. And so it's going to be a ratio of greater than 1. So it's a common way to look for positive selection. There are a lot of problems with it, but it's common. So why on earth is so much of the genome in malaria looking like something like positive selection? Is it really positive selection? What's going on? So under the life cycle model, I'll argue that it's actually completely neutral and expected. So we have this tiny population size, and under 1970s, 1980s nearly neutral theory, we can go back to one of the base equations of population genetics and see that the nearly neutral boundary of genes that act as if they are neutral depends on population size. So in a small population, selection is much less efficient and allows these slightly deleterious mutations to drift to high frequency because it's not able to kind of purify them out. And so malaria, with its tiny effective population size, has this dramatically expanded nearly neutral zone, allowing deleterious mutations, which are more likely to be that non-synonymous like numerator, to drift to higher frequency. And there's one more impact that we have to see to look at for in empirical data, because it's not just the expectation of pi n and pi divided by pi n s that's changing, like the ratio that's changing, which is, say, here we see an expectation and maybe it's like 0.7 instead of 0.2, but that's still below 1. Like, how do you get above 1? And I think that that's just statistical artifacts of sampling. Because not only, it's changing not just the expectation, but the variance. When you have fewer sites, it's going to change the variance to be higher. So we looked at empirical data from 700 samples from 4 populations in malaria gen, and just a quick table. We're still working on making these look nicer. But across the 4 tables, a few things to highlight. So one is in blue, genome-wide, you're still getting the signature of, most things are purifying selection. It's not too crazy. Genome-wide, you have something like 0.4 is the ratio of pi n to pi s. But if you're going at this per gene level, you now have way fewer like informative sites per gene, and you're getting a mean per gene that's closer to 0.8. And so it's not surprising if you're already at 0.8 that with a little bit of sampling bias, and here the variances are calculated, and this coefficient of variation is like the ratio of the mean and standard deviation. And when it's above 1, that means that it's like really a spread out distribution, is pretty high. And by chance, you end up with these numbers that are on the order of like 20, 25% that are over 1. You don't have to invoke selection at all. So the excess of genes with pi n pi s over 1 is really a combination of low effective population size expanding this nearly neutral zone, but then again, low effective population size expanding the variance as well without any need to invoke selection. So what's the expected genetic variation of a malaria parasite? Well, we developed this 4-stage model. It's a very simple way to think about diversity in malaria. And we think that it helps us build intuition about the impact of various parameters on genetic diversity. You can play around with, well, if we change this, how does it impact? In particular, it identified like certain like functional studies that might be really helpful. Like we want to get a better estimate of, say, the bottleneck size in malaria and how much variation that is if we want to understand and model malaria better. But, it doesn't really matter if we get parasitemia right. So that's okay. It can help direct these studies. It also can help us interpret common summary statistics in these empirical studies, with the example of Tajima's D being a negative in Tajima's D being induced by the life cycle and sampling scheme. And in particular, sampling scheme, I think, is really underappreciated when talking about empirical studies, not necessarily population demography. So our interpretation has to be like pathogen or organism specific and not just about the base theory that was developed for this statistic. So with just the last couple minutes, I want to mention, because as I said, I do see this as a little bit of an introduction to being at UCLA, a whole other line of work that is kind of in this vibe, but I'm not talking about yet, but would be great if there are people who are interested in rotations or just want to hang out or collaborate or anything that we've been thinking about, where we have primates as a model system for, as well as a reservoir of human infecting malarias. And so we have on the order of 20 different malaria parasites in the whole phylogeny of malaria. Only a handful of them regularly infect humans. Those are in yellow. A couple more can be forcibly infected. People used to do studies in the 1960s that we don't do anymore, but and it kind of spans the whole phylogeny. So it's not that kind of all human malarias are closely related to each other within the phylogeny. And I'll mention that this tree is incredibly diverse, where they have different genome sizes that are about like 1.5x different. AT content goes from like 80% to much more normal, like more around like 50% down here. So these are incredibly diverse. Why do some malaria parasites infect some primates and not others? And so that's kind of been an ongoing question that we're starting to look at from a more empirical perspective, and particularly thinking about, where did the slide go? A case study in howler monkeys in Brazil, where we, me down here, my crazy hair, see a lot of opportunity for overlap in zoonoses. So we were staying in a town, Teresópolis, that's about 100, 200,000 people. And we walked down a main highway for about half a mile and got to sample the monkeys. They're like right there. And there's a lot of standing water, opportunity for overlap between, what you need for zoonosis is a mosquito who's willing to bite both a human and a monkey in its lifespan. So we are trying to do a whole bunch of empirical data collection, sequencing the howler monkeys as well as their parasites. We've made new long-read assemblies. We are just starting to analyze a whole bunch of high-coverage short-read data as well, looking at both their kind of larger evolutionary genetic questions, karyotype evolution, as well as the malaria-specific questions. Where did that go? Importantly, one of the cool things that we're just looking at is whenever we do the sequencing of the howler monkeys, we also are doing PCR tests and then hybrid capture to get their parasites. And so far, about a third of the monkeys, at like 500 or so that we've tested, are malaria positive with a variety of different malaria species that we're trying to get whole genome sequence data from now. So that's a little side. So coming back to our main questions. The kind of things that the main things that we've been thinking about in the lab that we want you to walk away with is that adaptation to malaria has profound but also ongoing consequences for genetic variation in human populations. We need better tools to interpret this, both on the empirical genetic data, but also on the theory perspective. Longer term, we really want to see wider variation in primate and plasmodium pairs of genomes and other information, phenotypic information, presence absence data from ecology, which can inform human malaria susceptibility, severity, vaccine targets, and zoonotic risk. And I will end there to leave time for questions. I want to thank you for listening, thank my collaborators on these projects, current and former lab members, who all, came with from Duke and our funding. Yay, we have federal funding.